Namaste to all students. Myself JV and Dr. Khushbu Verma from Faculty of Agriculture and Veterinary Science. Today we are going to discuss in detail about the G protein signaling, and this session is related to cell biology course of program Beta Cavity and BSc FST second sem. In previous session, we have discussed about various types of the cell signaling, intercellular and intracellular signaling. We have also discussed about the types of receptors, in which we have already discussed about the basic structure of the G proteins receptors. But in this session, we will discuss about the G protein mediated signaling in the detail, and what are the secondary messengers which has been regulated from the G protein signaling and how they further do work, and what are the final products of their signaling. In this session, we are going to discuss in detail about the G protein signaling and their final products. So now come to the first of all concept of the signaling. The process in which cell sends the extracellular stimuli through membranous or intracellular receptors, transduces the signal via intracellular molecules, and thus regulate the biological function of the cell. There are two types of a signal which can create the signaling process, or it can trigger the signaling process. Physical signals and chemical signals. In case of a physical signals, light, UV, heat and volume comes, and in case of a chemical signals, there are various examples like hormone, neurotransmitter, growth factors, cytokines, odor molecules, ATP, active oxygen species, ROS, drug toxins, etc. They are they are the classification or of the chemical signal. So now come to the mode of the function of endogenous signal. As I have already told you, there are four types of signaling: endocrine, paracrine, autocrine, and synaptic. In endocrine, act on a far away organ via blood circulation. In paracrine, act on a nearby target. In autocrine, cell act on itself after secretion. And in case of a synaptic, presynaptic to the postsynaptic signaling happens. Now come to the primary pathways of the cell signaling. There are two types of a pathway: G protein mediated pathway and non-G protein mediated pathway. So first of all, we are going to discuss about the G protein mediated pathway. It happens by three categories like adenylyl cyclase mediated pathway, phospholipase C mediated pathway, and small G protein mediated pathway. G proteins coupled with the membrane of 7 alpha helix transmembrane domains of the receptor superfamily, which are regulatory proteins that act as a molecular switches. They control a wide range of biological processes. Now come to the classification of the G protein. So there are two types of a protein: large heteromeric G proteins which contain high molecular weight G protein, trimeric GTP binding regulatory protein. Second type is a small monomeric G protein which includes only RNAs. Now come to the regulation of the G protein activity. So in G protein coupled receptors, extracellular portion of the receptor provide messenger binding site and a cytosolic loop connecting the 5th and 6th transmembrane alpha helix is specific for G protein binding. Receptor occupation promotes interaction between the receptor and G protein on the interior surface of the membrane. This induces an exchange of GDP for GTP on the G protein alpha subunit and disassociation of the alpha subunit from the beta and gamma heterodimer, because G protein has been made up by the alpha, beta and gamma. Depending on its isoform, GTP alpha subunit complex mediate the intracellular signaling either indirectly by acting on effector molecules such as adenylyl, cyclase AC, or phospholipase C, PLC, or directly by regulating ion channels or kinase functions. Here you can understand G protein mediated signaling by this flow diagram and sequential explanation. When G protein is in resting stage, so G protein has three subunits, alpha, beta, gamma, and alpha has bind to the GDP molecules. And now this is the complete structure, and this is inactive. When ligands come and bind to the receptor, G protein releases basically G alpha, and this alpha protein releases the GDP and acquires the GTP molecule. Now, G alpha with GTP separates from the beta and gamma subunits. This separated subunit of G protein further inhibit the target proteins and initiating the signal transduction events. Here, G protein basically mediated the signal transduction pathway in three types. First one is the adenylyl cyclase mediated pathway, second phospholipase C mediated pathway, and third one is small G protein mediated pathway. What is G protein mediated pathway? So basically, interaction of alpha subunit of G protein with adenylyl cyclase. Here you can understand this connection by this flow chart, where this is the binding site of alpha subunit of G protein, and this one is the active site which has been closed, and binding and induced fit. This is adenylyl cyclase. When this alpha subunit which is activated, they bind to this adenylyl cyclase, and after the activation of the adenylyl cyclase, it creates the cyclic AMP molecules. They change the ATP into the cyclic AMP. So alpha subunit basically changes the shape, weaker binding to the enzyme, departure of subunit enzymes revert to the inactive state after the completion of work. And after the completion of work, this alpha subunit further again recombine with the beta and gamma dimer to reform G protein. In the second type, which is adenylyl cyclase mediated pathway, here you can simply understand this is the adenylyl cyclase. This one is a G protein. When it binds to the adenyl, especially alpha subunit in activated form, when it binds to the adenylyl cyclase, it creates the cyclic AMP. These cyclic AMP further bind to the CAPK, protein kinase A, which can phosphorylate the REB, which is binding protein for cyclic AMP responses element binding protein. This is the active form of CAPK, which bind to the CREB, which initiates the gene expression or gene transcription. This is the CRE, which is CAMP response element in the DNA. When it has been activated, so it creates the basal transcription machinery. So this is the adenylyl cyclase mediated pathway. Next one is the phospholipase C mediated pathway. Here you can simply see this is the phospholipase C, which is inactive in form. And this one is the receptor, and this one is a G protein, which is inactive. When ligand comes and bind to the receptor, it activates the G protein, and this G protein replaces the GTP from the GTP and separates from its beta and gamma subunit. Now this separated alpha subunit with the GTP, it bind to the phospholipase C and create it into the active form. This phospholipase C molecule change this PIP2 into the IP3, inositol triphosphate and DAG. Basically, IP3 is released after the activation of IP3, it is released into the cytosol when it triggers calcium ion release. On the other hand, DAG remains into the membrane when it activates and create the protein kinase C. Next type, which is G protein mediated pathway, which is phospholipase C mediated pathway. It create these kind of a secondary signaling pathway, and this one is a G protein linked receptor. This is a G protein with GTP. It activates the phospholipase C, which degrade PIP2 into DAG and IP3. These are the secondary messenger, and this IP3 further go and bind to the IP3 gated calcium channel, which activate this channel, and this channel basically present on the endoplasmic reticulum membrane. After the activation of this channel, endoplasmic reticulum releases the calcium ion from ER lumen to the cytosol. After the releasing of calcium ion, these are also secondary messenger. These secondary messengers, further 4 molecules of the calcium ion bind to the calmodulin. After the activation of this calmodulin, it activates the various proteins and further go for the cellular responses. So that's how phospholipase C mediate the signaling pathway. What is the role of the signaling? So it stimulates the cell growth, regulate the ion channels, change in cytoskeleton, and elevation in the cytosolic pH and secretion of the proteins. So these are the cell events which happens after the signaling completion. Now come to the perspective of G protein signaling role in plant and animal system. In animal system, role of G protein signaling is well known in prescription of light, odor, certain taste, hormones, and neurotransmitters. According to the recent studies, G signaling is at the heart of many plant photosynthesis physiologies and of agronomic importance such as pathogen caused disease resistance and harvest index. So in future, translation work will certainly improve the agriculture process. So this is the sum up of this session, and this session is powered by digital version 2.0 Jyoti Vidyapeeth Women's University Jaipur. And if you have any query, then mention in the comment box. I will resolve it. Thank you.